I guess I'm able to share the screen here. So I've prepared a little introduction to this session, but then I hope we can open up just for questions by whoever is asking questions. And I'm not sure, Coca or Carlos, is it just, I guess everybody can unmute. They can put questions in the chat, but they can also, I don't know, make their voices heard, I guess.
Yes,
Yeah.
Seb,
Yeah.
depending on the size of the
Yeah.
room,
Yeah. Let's
how many
see.
attendees, but I
Yeah.
think we can either direct forward the questions to
Yeah.
the
Yeah.
question
Yeah.
and answers, or
Yeah, and maybe you can help me a little bit. I'm an old professor handling all these technical things, and I may miss questions in the chat or whatever. It's not that I on purpose want to diss anybody. It's just happening, I think, because I'm not capable of handling everything at the same time. So if you just be a bit patient, and if I seem to ignore you, just I guess kick me under the table somehow on the distance and get attention. Okay, so maybe it's yeah. I can start sharing a screen so that we start to see at least the title page of the presentation that I have. And then what I've done is I sent an email to you guys very late yesterday that I actually took screenshots of all your slides. So if we get questions, I think, and it will be helpful to look at slides. I have them on my screen. It will be much easier that way instead of switching back and forth between different people sharing screen. That was my idea. So let me start doing this way then. Okay, so now you see something else than I want to show. That's what I want to show. Let's see. Okay. The downside is when I show the full screen like this here, I don't see anybody view anymore. So if you want to kick me, you have to do it by raising your voice. Okay.
I can see your screen and I see a bar of all the participants. So there must be a way in which you can see a bar of participants.
Yeah. No, you do it, but I at least when I use it in teaching, also, I don't know. I even make maybe I should have the bar left, but then I don't see my slide. Oh, okay, so I can see participants, but somehow I don't see the chat anymore. So I would have to go on things here. But bear with me for a little while, and let's get started simply. Okay. So this is the name of our session here. Before I get serious, I want to have a little warm up. So those you can see me, you may notice that I'm wearing a kind of Corona haircut. That's what everybody does nowadays. At least is at my age and wants to be a little bit careful. I'm also sitting in Umeå, so we have lots of snow out there. I know, Wolf, when we had you over a few years ago, you love to see the snow, even in May. I can show you we had tons of snow. So this is what it looks like here. Let's see if this works. Yeah. You see, this is what we do. So this is what I do for exercise right now, shoveling snow. And we really got tons of snow. So this is in my apartment building. We are trying to save our garage building from breaking down under the snow. And this is really unfortunate. I mean, we just, before we left the general session here, we saw these nice pictures from down here. We could all be here if the situation would be different. Maybe not now, but we would have been there in June. And instead of shoveling snow, I would have liked to try this. Yeah, we certainly would have done a lot of this, I believe. But it's pretty amazing. When I was Googling for beachfront bars in Natal, I got this up, for example. And if you look carefully on the bottle, you get back to the reason why we are actually sitting at home and are shoveling snow. That's what's going on right now. Yeah. So let me get now serious about our session here. The reason why I, together with Francisco, suggested this title here is that the whole thing about feedbacks is what got me interested in modeling a long time ago. So I'm an aquatic ecologist with empirical training. but I have very soon realized that a lot of, when you read papers, you have in the introduction often these kind of linear reasoning. When A happens, then B will happen, and then C will happen, then D will happen. But this is not really how it works in dynamical systems because of these feedbacks. And I want to illustrate this with a couple of theoretical examples. So, for example, actually, I think I can have a pointer here, laser pointer. Yeah. So feedbacks can buffer the impact of environmental drivers, or they can reverse it. And this is a very simple model I'm showing here as a cartoon where I look at nutrient enrichment in, let's say, producer-grazer system. Could be algae and Daphne or something like that. If you run this model to equilibrium at higher levels of enrichment, what you're seeing is that the algae don't change in biomass at equilibrium. So this is a very good example of how environmental change is actually buffered. And this is the whole reason why biomanipulation, for example, can work in some systems when you try to benefit the grazers of the phytoplankton. Now look at the same food web when you now add a fish on top of that, one that can be herbivorous and planktivorous. And now look at the middle. In the previous model, we see that it's this trophic level that responds to the environmental driver of nutrient enrichment in a positive way. But if you have an integral predation system, which you have here, at least in theory, you would expect the opposite now. That all of a sudden we see the reverse, that the middle level declines, and it may even go to extinction at sufficiently high level enrichment because of these feedbacks, that when you increase nutrient enrichment, you're actually benefiting at the same time this top consumer here that has in total then a stronger negative effect on the middle level than the increase in resources. Another example that's quite prominent in this session here is the thing about positive feedbacks. Here I've taken an example from my own slides in my talk. They can promote alternative states. So when you have this kind of loops, where if you just go along the state variables and the arrows and multiply the signs, and then you get here minus times minus is plus and everything else is plus. So you have a positive feedback loop where nutrients may benefit the pelagic algae, but benthic algae may reduce the nutrient flow. And then what can easily happen is that you get maybe either this state or this state of the system, and it could be going back and forth between depending on environmental conditions. And what it definitely means is that because of these positive feedbacks, how a system responds to an external driver like a nutrient input may very much depend on what kind of alternative state the system is in at the beginning. So this is a figure from Monkey's talk. I've borrowed a few from slides from other people's talks here. And then this is from Wolf's talk, just to illustrate with his food web graph that feedback loops are everywhere. And I'm just picking out one of them that he also highlighted in a way where we have a new actor in the system here, or at least something is changing because of environmental change. And that could be that species 2 here is evolving, or it's being harvested by humans. There could be a species replacement, that there was species 3 before, it has been replaced by species 2. And you see there's all these other feedback loops in the system. So it's extremely difficult to predict how systems respond. That is kind of what always has fascinated me with ecosystem dynamics. And I want to claim here that process-based mathematical models are one of the most powerful and versatile tools that we have to explore the consequences of feedback processes. So this is one example of a pretty elaborate lake ecosystem model, the PC Lake, that several of my fellow session members here have used in their research. And I just want to illustrate a few of the ways how you can use these models and to analyze. So this is from Alena's talk, who got interested in what happens when we have these submerged macrophytes, the plants, and there is actually periphyton growing on them, a component that was before missing from this model. So she seems to have introduced it or added it to this. And the one thing I just want to highlight here that you see again one of these situations where depending on how much shading there is initially or in which state the system is, whether there's a lot of macrophytes or a little, which is here, there must be a positive feedback loop here that is very important in the system. That's one thing you see here. Another approach is the one that I used in one figure in my talk, which would be a two-parameter bifurcation plot, where basically we looked at the effects of changing terrestrial dissolved organic matter, which is colored, which makes the water turbid and brown, and the sediment nutrient input to the system. And you see how, for example, in this case, phytoplankton and other components could react to this at equilibrium. And again, you see areas of alternative states, but you also see areas where there are very abrupt system shifts from a system that has very little phytoplankton in clear water, where there would be benthic algae dominating in my model, and then very abruptly with just a little bit of browning here, for example, then you move over to a system where you have lots of phytoplankton. And then finally, Annette used an approach where she looked at how local feedbacks propagate in space by putting lakes or lake compartments next to each other in the flow direction, and explored the consequences of that. And inside each of these boxes, you see the lake PC lake model that produces a lot of feedbacks internally, but some of that is then propagated by, for example, downstream flow. So here is a list of the 8 talks that were uploaded on our website. I hope that all of you had a chance to listen to them. I will actually not really go through them in any detail here at all, assuming that you have looked at them. I won't even read up the titles, but my plan is I will very briefly just go through them. And then we come back to this table here. And my suggestion would be that then we just open up for questions that all of you have, and going through these talks in this order first. So first specific questions to the talks, and then when we are through with that, then we open up for a more general discussion. That is my my suggestion. Yeah. Okay. So I start with Martin here. So he presented an analysis of basically determining the diversity and other things in a comparative study of lakes, natural and reservoir lakes. And the idea here was to make a prediction of how a system would respond to decommission of dams. So where a dam is taken away, I think from this lake here, that was the main question in his talk. Then we have Maria here. She was presenting a dynamic energy budget model describing the individual growth and life history of tilapia, which is a fish that I think has been introduced to lots of places in Latin America as a potentially commercially valuable fish. And she's exploring how especially availability of resources to this fish shape the life history, the growth, the reproduction, and so on, and compares it with data. So these 2 talks are maybe a bit unrelated to this theme of feedbacks I was mentioning earlier. Then comes a whole suite of talks, all of which have to do with the role of light as a potentially limiting factor for primary producers in lakes. And the first one here is Carlos talks. He specifically investigated the situation where reservoirs, or maybe also natural lakes, but I think many of them were reservoirs, that can have huge variations in water level, at the same time often very nutrient rich in the area that he was studying, where these changes in water level can affect the light environment below, and how that affects primary production and things like that, biomass of algae. That was one thing. Then we have a talk that also deals a lot with light by Monkey here. She was particularly looking into how traits of cyanobacteria, 2 of them, one of them is buoyancy regulation and another one is nitrogen fixation, how they could affect their performance in competition with other primer producers in the water column of lakes. And here the buoyancy regulation you can see could give them perhaps a competitive edge for light if they can float up to the surface. Then Alena was looking also at a competitive interaction between primer producers. In this case, they are not moving around, they are sitting where they are. So there are the macrophytes, and then there is the microscopic stuff that grows on them, which quite often doesn't look so microscopic anymore when they build huge biofilms. So she was interested in adding epiphytes to an ecosystem model, epiphytes on macrophytes, and how that system then responds to things like nutrient enrichment. Then there's my talk where I compare, or I actually develop a pretty simple model that also looks at competition for light and nutrients between two groups of primer producers. So it could be macrophytes and benthic algae, I call them here, and pelagic algae, or it could be things that float at the surface, similar to monkey's case and pelagic algae, and develop those and compare them with some data. Then we move up in scale. So this is Annette's talk, one slide from her. She was particularly curious about looking into what happens when we have both hydrological and nutrient loads that are either point sources or diffuse sources, or you have all combinations of the water, the hydrological loads being diffuse and the nutrients point sources or vice versa, and exploring this also with the PC lake model in arranging them in a sequence. And then finally, we have maybe the broadest scope of the talks that were uploaded by Wolf. So I have to tease you a little bit, Wolf. It looks like you were sitting back in your armchair reading books and then presenting them to us. But there was one figure that particularly, of course, caught my curiosity, which is this one here, looking at the interaction of socioeconomic and ecological systems, where you may have alternative states or things like that in both the behavior of humans acting on the ecosystem and in the ecosystem itself. I just love these kind of figures. And when they start to look that complicated, I always get a mental association to this beautiful sculpture, the Laocoon group, which is, I think, placed in a museum in Berlin. I've actually never seen it in real, but it's something that caught my attention already when I was in high school and just saw it in books. It's just wonderful. And, especially if you kind of flip it around, it almost, here, this beautiful line, you find it there. And here in the equilibrium point, you find this great expression of blissful suffering, I would say. Yeah. So great. I think I'm through. With giving very short introductions to the talks. Here's now the list of let's see, of talks, and I would simply open up the discussion in the sense that we start with if there are any questions that are specifically targeted at Martin's talk first, and then we go through Maria, Carlos, and so on. So maybe let me see. Let me get out of this. Yeah, okay. So I kind of put it like that because then I can fish up other presentations if necessary. So let me see. In the chat, is there anything? There's nothing in the chat. So anybody who is online now is just welcome to raise their voice and ask a question. Okay. Don't be shy. Let me see. Martin, I haven't welcomed you yet, but I hope you're present. Yes, you are. Okay, I just go through the list here. Yeah. Okay, Monkey, you raised a hand. Okay, yes. Yeah, please.
I may first ask one question. Hi, Martin. Thanks for your talk. Very interesting talk. And maybe it's a simple question, but from your presentation, I get that the water level fluctuation can enhance the production, for example, for the lakes, in terms of, you mentioned it can bring oxygen to the sediment, but maybe also other factors. So my question is then, what do you think about the positive effects and the negative effects of the water fluctuation you mentioned in your presentation? Thanks.
Well, the positive effects would be this higher amount of available energy for the benthic macroinvertebrates, especially for the filtering collectors, because besides the higher oxygenation of the sediments, it would increase the amount of organic matter in the water column, because it would create more currents, it would revolve more the sediment, so it wouldn't deposit more. And it's hard to find a positive effect for water regulation in terms of ecology. I have some friends that works with macrophytes. They see it makes the pattern of the macrophytes more clear, but besides that, it's not very positive to regulate the water level of a naturally fluctuating lake, especially in shallow lakes, because it would make it poorer, it make it with let's say it make it easier for it to accumulate too much organic matter and then become a wetland and then a dry ecosystem. So if the lake is naturally fluctuating, all its process would be adapted to this fluctuation. So to stop this fluctuation would be detrimental for the ecosystem. And this is what I found in this study.
Okay,
yeah,
thank
thanks.
you. I had a little question in the same direction, just for understanding. So, which is very shortly answered, I think. I wasn't quite sure. Did I understand correctly that the natural lake is actually fluctuating more on an annual basis than the one that is regulated?
Yes.
Okay. Okay, because I'm just curious, because when you look at the same situation up in Sweden, for example, where we have lots of dams for hydroelectric power, it's the opposite. The natural lakes fluctuate not very much, but of course the hydroelectric power plants can fluctuate a lot, especially in winter when there's not much water coming, but electricity is still gained through basically letting the water run through.
Usually the case here too, but since this is a I forgot the term. This then does not work anymore. It's almost so it does not have this fluctuation based on the use on the hydroelectric production. So it's really just a artificial lake now, a very shallow artificial lake. And this lake is not the reservoir lake. It's a natural lake that is connected by a shallow channel to the reservoir. So it's this that is stopping the natural fluctuation.
Okay, thank you. Yeah. Are there any further questions to Martin? If not, then let's go to the next presentation by Maria, the tilapia life history. So please, any questions here? Okay, there's something in the chat.
I don't know how to raise my hand. How do you do that? But can I ask you something?
Yes, go ahead, please. Yes, Yeah.
So, well, it's a remark. So you said at the beginning that the first two are less connected with the feedback thing, but I think the model that Maria works has a strong feedbacks and alternative stable states and all that kind of stuff. So there seems to be a misinterpretation. The feedbacks are within the population growth. So you can have these stunted populations, and can have the population with lots of adult fish and a little small amount of small fish and so on. So although
yes,
it focuses on a different, not on ecosystem, but on autogeny and so, but it bears many resemblance with the systems that you focus on in terms of feedback mechanisms. So I thought your disqualification that it was not about feedbacks was not completely appropriate for that talk.
Yeah. So maybe I misunderstood this then, Maria, and then you can correct me, because the impression I got is that you were basically looking at the life history and growth patterns in individual fish depending on food abundance, but there was no feedback on food abundance. So I think you were treating, you're basically looking at the asymptotic size of your fish in lakes or in other data and dividing it by the maximum size that is possible at all. And that was your estimate of let's say, growth conditions. And then you looked at how your growth curve looked like. But I didn't see any feedback there. And I didn't really understand, since you, in your introduction, you presented very clearly that there are maybe not clear stages, but there's a shift in diet from very small to large stages. Which must mean that they use different resources. It was phytoplankton in the older stages and the larger, and zooplankton in the smaller. But you used a single value of F when you looked at the growth patterns, and that didn't go together totally for me with that they use different resources and that you could get stunted populations in one case and not stunted in others. Must to some extent depend, I think, on the abundance of both zooplankton and phytoplankton. But maybe you can comment on that, Maria, please.
Oh yeah. Hello, everybody. My name is Maria. You already have seen. In fact, there's a problem. There's not so much feedback here as expected, right? But maybe it is, well, it was not clear during the presentation. It was really short, and the work, the manuscript is a bit larger. But the aim of this work that I'm present here is that we have I started with this work here in Brazil, and here in the eutrophic lakes, we found two different situations here, where we found the situations where the population is small, the population is small, but we have big tilapias, and lakes where the population of tilapias is really large, is overpopulated. The lakes are overpopulated, and the tilapias is really small. Okay, so I don't like you, you don't like me. So in this work, we try to start to model this, what was happening. Somebody's with the microphone open.
Yeah, we have someone, there's noises in the background, so those are not
I
talking. Okay.
So maybe it was not clear in the paper because here, not just here, because I did a review on the literature. I'm going to ask these questions here on the chat. But we did a literature review and the data that we have here in Brazil. And the feedback is that in the paper, is that if I have a eutrophic lake, in fact, it's not so much a feedback, but if I have eutrophic lake, the fact that the tilapias have this difference in the diet during the life stage, and tilapia, adult tilapia is it's algae. It's on the algae, while the juvenile tilapia, it's on the zooplankton. If we introduce the tilapia in a eutrophic lake, we have a lot of food for the tilapia, for the adult tilapia, right? And the adult tilapia reproduce a lot, stay reproducing in a really large amount, and this increases the eutrophication in the lake. While for the zooplanktivorous, for the juvenile tilapia, it's decreases the food for the juvenile, and two things happens. Increase the eutrophic state of the lake because they are there. I forgot the word here. They are putting more nutrients in the lakes, and they decrease the amount of zooplankton. The small ones are eating zooplankton at the same time. The small amount of zooplankton that already have in the eutrophic lakes. And at the same time, they are producing more and more fingerlings, more and more tilapia, and increasing the eutrophication of the state, and at the same time increasing the size, decreasing the size of tilapia, because there is no food for the juveniles. So there's this relationship. If I have large tilapias, if I have eutrophic state, tilapias reproduce more, and at the same time they decrease the size of the juveniles and decrease the size of the tilapia in these lakes. In this lake, we don't know, most of the lake here, they don't have macrophytes, and we don't have, in the literature review, we don't have the description if the lakes have macrophytes or not. So it's, in fact, I stay almost more than a year looking for the literature, but they don't have data from submerged macrophytes. They don't have data of phytoplankton, just a few data of that. They don't have data of zooplankton. So it's difficult to model the food for tilapia. Is really difficult to model this, and because of this, we just model it now based only on the food results for the adult and not for the juvenile for now. I don't know if I was clear during the
So maybe I can. First of all, I want to make clear, I didn't want to disqualify your talk, Maria. I hope you didn't get this idea. I'm enchanted that you chose our session here. And I liked it a lot. The thing I didn't quite understand is, I would expect maybe a standard population in a situation like you just described, where you have very little zooplankton. You have, but you have then lots of phytoplankton, which might be good for the few adults that are there. So they will produce a lot of offspring, a lot of eggs, and so on. So there will be a lot of, let's say, pressure on the young stages because there's a high fecundity. But then these younger stages will compete very strongly for the zooplankton and then show slow growth. I would think, and then you may get a stunted population. But wouldn't the few who make it to the adult stage that have become big enough then have a paradise and grow very, very fast and become very big? Or am I misunderstanding things? Because the phytoplankton, which is the resource of the adults, should be quite abundant then if the zooplankton is suppressed.
Yeah. Yes, and then zooplankton is depressed. Yes, it's exactly this. Yes.
Yeah.
Maybe it was more clear because this is the first paper for the model, and the next one is the population dynamics that I could not show here. So maybe it's something was not understand here. And he said that I should have put in the fishing in lake system. Maybe yes there, but okay.
Yeah. Okay.
The fishing session.
Yeah. So then I see there's in the chat there's a question. It's a pretty long text, but I think if I boil it down to the questions, there was first I just wanted to confirm whether tilapia you looked at always invasive to the lake system. And then second, I want to ask if in your literature review, did you find anything on tilapia feeding on submerged macrophytes in our lake? So it's a person from Madagascar who's asking the question, I think. In our lake, it appears that the introduction of tilapia may have caused a shift in stable states, resulting in the loss of submerged charophytes and reduction in water lilies.
Yeah. No, in the papers that I looked for, we don't have any nothing about the feeding on submerged microphytes, but maybe that depends on the kind of tilapia. This Nili tilapia doesn't eat too much macrophytes, more phytoplankton and zooplankton. Maybe depends on the kind of tilapia that was introduced. And no, in the paper we have data of tilapia in both lakes where she was introduced, and lakes where tilapia was native, there was already there. It's a pattern for the world, but this that you talk about, that tilapia may have caused a shift in stable state. We have also something like this here because of this the input of nutrients that she puts in the reservoir, because she suspend the soil on the reservoir also, and because of the high reproductive rates. Here we have this, and we here the tilapia did not change the reservoir state for a more eutrophic state when we have the predator, but eating the juveniles. If we have the predator, we have a clear water state. But if we not have the predator, we have this absence of macrophytes, we have eutrophication and the other things.
Okay. Yeah. Okay, I saw that Wolf has learned how to raise a hand. So please, Wolf. You have to unmute yourself. Wolf, you have to unmute yourself.
Did I unmute myself? Yes.
Yes.
It's a very short. I wasn't the first to raise the hand. I said, but I think it's not useful that we look at your slide all the time. So maybe we can just that we look at each other better during the conversation. So if you can.
Yeah, sure. No, that's
Yeah,
easily
okay.
done. So let me actually
because
just,
you can just list and name the next person, and I think then.
Yeah. So I can
use the non-verbal communication.
Well, I guess let's see. I guess I have to stop share. That's what I
exactly.
think. Here we go. Yeah, that's much easier. Yeah. Okay. Thanks. And then Koka had raised his hand.
Yes. Thanks for raising this issue, Sebastian. I think you are right when you say that in this presentation, the feedbacks in the model are not so well explored, because the intention here was first to parameterize the model, and then there is a second paper by Maria where she explores the dynamic of tilapia's population. And our intention with this model is actually to find out what should be the fishing predation pressure on tilapia population to get a more to increase the fishery yield of tilapia, at the same time decreasing the negative effects of tilapia on the water quality. That's the goal with this model. But the population consequences of these feedbacks at the population levels are not really so clear in this presentation of Marcolina, because she's presenting the huge effort she did to parameterize this model. Okay,
thanks. I suggest that we go on now to Carlos, so we can at least get through everybody and then have a little bit of time left for a general discussion. So if anybody has a question addressed to Carlos, who was presenting about the water level fluctuations, please either raise your hands or see. The chat is not.
I have a question
Yes.
about Carlos' work, but not direct addressed to Carlos. It's just to all of you guys, because we work in systems that have huge variation in water volume. And we see most of our limnological models, the great PC Lake model and others, they assume a constant water volume. So I wanted to know, I mean, if Wolf and you guys are considering the possibility in future generations of lake models, as Wolf has put in his talk, to include water balance and water level fluctuations to better integrate the aspects of water quantity and water quality in water management, because we really need to do this. I think our models, assuming constant volumes, they might be useful for some types of systems, but the systems we work here, and considering climate change, extreme hydrological events, droughts, water abstractions, will all change very much the water volume and water depth. And I think this is something that we should consider with more attention. What do you think?
Yeah. Maybe I can respond immediately. I would argue that
Annette is raising her hands to respond to this, I guess, or not?
Yeah, I will give her the word, because my immediate comment would be that Annette has already demonstrated, you know, that hydrological balance is easy to include, and I think probably maybe I shouldn't forego your answer then, Annette. So let me unmute yourself and answer to it, please.
Yeah. So first of all, PC Lake already can do this. So there is a water balance in PC Lake. So you can change the water depth, you can change the volumes. I think the issue here is more that the model is mostly used for shallow lakes, where the water level was more stable than maybe other lakes that like the deep reservoirs, where you can have a sudden drop. So I think that is the main reason that we didn't try it, but I'm sure the model can do it. Yeah, but that said, so the model has the ability to have changing water depths, but I think the model is mostly focusing on ecological things and not so much on the hydrology. It is possible, but I think also it's a very simple way it's included.
But remind me, Annette, the study by Kong of Lake Chau is included water level variation, right? So we have an extensive study over 50 years that focuses on the interplay between water level variation and hydrology and vegetation over a 50-year period. So there is a long-term study on that.
So in that study, indeed, is for Chau Hu, where PC Lake was used to see what was causing the big blooms in that lake. Was it either the dam that was put in place that higher the water level but also made it more stable, or was it the eutrophication? And I think what was the main conclusion of that paper was that by placing the dam, the eutrophication problem started earlier, but it was eutrophication itself that caused the problems. So, but I can share the maybe the reference in the chat.
Yeah, and so maybe one comment I have on this is also I didn't quite understand what was going on in your model compared to the data, but the data you were showing, Carlos, they indicate that there is actually a huge drop in water in volume towards the end of this period. So it's a huge effect, and my guess would be that if you have a situation like that, then maybe a model that is a 1D model. I guess the PC Lake is usually assuming a water column, not different depth, you know, that you have shallow and deep areas. So maybe you need to go to something that includes variable depth also, because your area will probably shrink. So you will have areas that will be shallow initially, they will be dry later on, and that kind of thing. So the morphometry of the lake may or may not be important. That's something to explore, I guess, with maybe not a 3D, but you can do a 2D model or so, which I've been starting to work on with a PhD student. But other than that, I mean, the idea that the water level fluctuations that affect the light environment should be potentially important is quite obvious, I think, from the system you have been describing as a very nutrient-rich system, high turbidity and so on, where light is probably a limiting resource. So I think this is a very useful approach you're going to. Then I had a few more detailed questions because I'm not understanding a few things, but that's something we shouldn't be discussing here, I think. So Koka and Carlos, if you are interested, we could set up a Skype meeting or a Zoom meeting at some other time, and I can ask my detailed questions about this if you, okay. So Dianeke and Monkey, I think maybe let's start with Dianeke. Maybe even the Monkey was first. I'm not keeping track really, but please go ahead, Dianeke.
Thank you. Yeah. I enjoyed watching your video, Carlos. Maybe I missed it, so it's all about changing the depth, your model, but I missed how you model the hydrodynamics of your system. Like, how do you include the effect of depth on your ecosystem in your model?
Okay, we made a water balance, and we take the volume and we divide it by the area, and we found the average depth in the reservoir, and we include this depth in some parts of the equations, and also the volume. We include this. We have some. We made some modifications in the equations, including trying to include this volume variation and depth variation.
Okay, then Monkey, please.
Oh, yeah, okay. I think this is for me more a comment as we discussed multiple questions. So I was thinking, because you mentioned that Carlos' model is a 1D model, so it has kind of a dynamic attenuation of light through the depths, which I think this is the difference between PC Lake and the Carlos model. So although PC Lake contain the water balance, but the kind of light availability doesn't really change when the water depths change. So that is what I think. Maybe yeah, maybe Annette can comment on this later. But this may be the difference that I can see between the 1D type model and the PC Lake 0D type model.
Okay, now let's see. Wolf raised his hand. So Dianeke, your hand is still up, but I think, or no, okay, yeah, they don't disappear after a while. But please, Wolf, yeah, unmute yourself, please.
Unmute. Annette can talk further on this, but I think the light availability goes down with depth because PC Lake uses a calculation to calculate the average amount of light over the column, which goes down as depth increases. What it can't deal with a situation where part of the phytoplankton community is light limited and part of the phytoplankton community is nutrient limited, which can occur in situations where in the deeper parts the system is light limited and in the upper parts it is nutrient limited. So I think the phytoplanktons and the plants as well are either light or nutrient limited, because there is no vertical description of the light gradient, but the light level on average does respond to depth. Isn't that the case, Annette?
Yeah, so yeah, indeed. So there is a Lambert-Beer equation in PC Lake where light decreases over depth. But indeed, so in the end, so the average light in the water column is calculated. And that means that, yeah, either it's low or it's high, but it cannot be everything in between. Yeah, so, or yeah, it can be, but I mean, like it's just one number. Yeah, so I agree with Wolf.
Okay.
But I'm not sure how the model of Carlos does this. So I'm not sure if that is indeed the difference or, yeah.
So maybe I should push this forward now so that Monkey gets a chance to get questions. So now we move on to Monkey's talk. So are there any questions? Or there's a chat, let's see. Okay, you posted a reference here. Okay, great work, Carlos. Congratulations, someone posted here, in case you didn't read it yet. So please, questions to Monkey. Okay, yes, please go ahead, Tobias. Yeah.
Interrupt, sorry. Really nice presentation, Monkey. It's nice including the functional traits of the Swan bacteria. It looks really great. I was wondering, you did your hysteresis plot, the bifurcation analysis. Do you think that how do you evaluate the sensitivity of nutrient load reduction scenarios with PC Lake Plus if you apply this in a management perspective? So, for example, I know PC Lake has been applied in management to try and set loading thresholds. So now you showed that this has changed for, I guess, a default lake. So how would you evaluate that if we go further on, maybe include other functional traits and stuff like that? How would this affect the when you apply the process-based model in a management perspective?
Yeah, I'm not sure whether I understand it correctly. Do you mean that what's my suggestion about how to evaluate the effect of other traits in terms of
Yeah, sorry for not being specific, but I was just curious to see, like you showed that you included new formulations and new descriptions of the phytoplankton, and this shifted predictions in your model. And when this model is used in management, I guess this will have an effect. So I'm curious to what your thoughts are on how robust or sensitive the model is when looking at thresholds for the formulations.
Yeah, okay, I get it. Yeah, so this model, I think this study provide a kind of option for the eutrophication management that they can choose to include these traits. So it's also, so for example, if you have a lake with the cyanobacteria floating on the surface and you know this species can fix nitrogen, then you use these functions and then you know that how much nutrient loading threshold will change in your case. And if you don't have this type of species, then you just exclude these functions. So there is a turn on, turn off options in the model. Yeah.
Okay. Walt has raised a hand. You need to unmute again. You need a personal trainer for this.
Yeah, thank you, Sebastian. You're doing a great job. It's a very complicated topic, I think, a very interesting topic that Tobias raised, because I think Manchi's answer, I like it in the sense that indeed, if you have a lake where there is no floating layers, then you don't need a model that includes floating layers. And when you have a lake where there are floating layers, you want the floating layers in the model because that's a real ecological phenomena that then affects the critical nutrient loads. But we had another example where we worked with chironomids. We introduced chironomids in the model, and that was a study with, I forgot his name, but from collaboration with Germany, and it says more about me, about the guy, of course. And he, introducing the chironomids in PC Lake had a huge impact on the critical nutrient loads. But what we learned is that we had omitted them first, but the grazing activity of the chironomids was actually done by the Daphnia in the model that were kind of over-parameterized in their grazing capacity. So what we call Daphnia took care of many other grazers in the model as well. When we introduced the chironomids separately, we had to take that grazing part out of the Daphnia component. Otherwise, we got just an unrealistic level of grazing in the model. So because the model is partly top-down calibrated to field situations, and it doesn't include all components, you kind of push things that are not in the model into components that are in the model, because otherwise the model can't fit the field data. In that case, if you keep adding stuff, you get a model into an unrealistic behavior. So a new phenomenon like floating, yes, then you may trust that indeed the critical nutrient loads change. But if you start just adding complexity of the model, you may lead to situations that were first aggregated, and then you get overlap between compartments that do the same thing and actually do too much. So it's very delicate ground indeed, the sensitivity of the critical nutrient loads to model additions. And we were struggling with that ourselves. Maybe Annette also has more about to say, but that's,
yeah, she has raised her hand. Yeah.
Yeah, so I think it's maybe a third perspective on this, but indeed, I agree with Wolf. You introduce something, in this case, floating or nitrogen fixation. And in that case, I think the absolute values of the critical nutrient load first needs a calibration to really trust them. But what it does say, I think, is the direction where the critical nutrient load will go to. And I think this could help management, actually. So if you have floating algae in your lake, for example, and you can see then from the graphs, if this floating algae can be managed somehow, what will it do with the critical nutrient load in your lake? So I think, for me, that is kind of the valuable information you can get from.
Yeah,
okay.
That was the same we had with the temperature study, right? We did a fairly theoretical temperature study, but it shows that the temperature increase from climate change would decrease the critical nutrient load. So that's also gave a direction rather than that you claim that you can predict the quantitative sensitivity of the model.
Okay, thanks. So Tobias, you still have a hand up. You have another question, or is it? No, you don't have to. Okay, so maybe I have a final question then. So I guess I misspelled you or mispronounced your name. Manchi, is it that what I should say? I'm sorry.
That's yeah.
Okay, yeah. So the one thing I was curious about is actually that you get this change in where the shift in the hysteresis curve is when you have the floating and two fixing algae, but I don't think in the presentation you gave any explanation for why the shift occurs, and also why the levels are higher and lower at different points. I think, at least, I may have missed it, but I think you only described it, and I got, of course, curious about why would that happen?
Yeah.
Especially maybe why the shift, you need a lower, I think you needed a lower threshold now for getting back to the clear state when you had these floating cyanobacteria, please.
Yeah. Thanks for the question. I think I do explain it in the presentation, the feedback loops in terms of the nutrient and light. So if you remember, there's some feedback loops figure that I present in the presentation. That's the reason, but I can explain that now also. So so there are different perspectives. So for example, for the buoyancy regulation, the buoyancy regulation influence more in the light side, and the effect on the nutrient loading threshold, it makes it becomes lower, which means the turbid state becomes more resilient to the nutrient reduction. It's because because the cyanobacteria can float on the surface, and it kind of starts endurance a very high light intensity, and then it's in a situation of the light inhibition because the cyanobacteria can be light inhibited, and that actually create a positive feedback loops that the increase of its biomass decreases light, but actually also benefit its growth, because the less light, the better for its growth. So
okay.
It cannot change the situation when it cannot float on the surface.
Okay, yeah, I had actually a different idea about what was going on. Couldn't it be that at the lower nutrient load level, when you already have a state of floating algae, and I think your system assumes to be it's nitrogen limited. So when you have a state of floating algae, then they can get their nitrogen from the air. Basically, they fix it, which makes them independent of what's in the water at this lower end. That's what make them more persistent. Could that be the explanation
that's
for
also
the
the
one.
reason.
Yeah.
Yeah, that's also the reason. So in the presentation, I only show one result, the scenario like with with two traits. Actually, in the paper that I published, I have different scenarios. So if you check the scenario that cyanobacteria only can fix nitrogen or only can float, they also have the shift nutrient threshold. So
okay.
yeah, that's also the reason.
Yeah, thank you. So then I suggest we move on to Alena. So anybody has questions to Alena, please raise your hand or raise your voice. Okay, maybe then I can start with a question to you, Alena. There were a couple of things I'm wondering about, but one was what I actually didn't really understand is in your hysteresis model, how Epiphyton can prevent macrophytes from recovering when there are no macrophytes on which, or maybe I'm misunderstanding, but on the way back, I guess the macrophytes were more or less extinct. So what does the periphyton grow on when there are no macrophytes? How can they prevent? Are they sitting on the bottom? Or I mean, a very simple question maybe, but I was confused.
No, thanks. So the thing is that what we did is we added a shading curve, which represents periphyton shading directly onto macrophytes to the model. So we didn't actually model the periphyton in itself. So that also means that the periphyton isn't linked to any of the other top-down or bottom-up controls. What actually drives the hysteresis there is that with less periphyton shading, at some point the macrophytes do, like when they sprout from the sediment, they don't get immediately overgrown, and then they could grow up again fast enough or far enough before they are hit by the shading again. And the other thing is that on the way back, I started with the conditions of a turbid lake. So there is also shading by phytoplankton, like the epilimnion phytoplankton included there as a part of the standard settings of starting with a turbid system. So that shading also already prevented for a large part the coming back of the macrophytes. But it is indeed one of the weaknesses of our study, what you point out here.
Yeah, so because the other thing I didn't understand, but that you partly answered it, is of course how exactly you model the shading, because it sounded to me like you're not really modeling biomass of epiphytes, so it's some kind of the shading function.
Indeed.
Yeah, and there were not really any units given to it that made it easy to understand how that works, whether it was mechanistic
no, it is indeed. It's just a proportional amount of light attenuated by.
Yeah. Okay, that's kind of what I guessed, but
attribute to periphyton, because that shading is only applied to the macrophytes and not to the other parts of the system.
Okay, thank you. Yeah. So Annette has raised her hand. So please.
Yeah, so thank you for your presentation, Alena. I was wondering, so we always tend to forget about this periphyton, but do you know how common it is actually in lakes in Europe or maybe around the world even?
Yeah, I think that would be the ideal question actually for Sabine. If
I can take over.
Yeah.
Please go ahead.
Send you a comment because you could have shown, if Sebastian would allow you to link to our Google Docs document. Of course, all these modeling studies always need a link to the real world. So what we will add to this study, but it didn't fit into this 12-minute talk, was a literature study on different real world periphyton dynamics. And we can show that you find in all sorts of lakes, from oligo to eutrophic and from all sorts of areas, that there is a certain pattern with like a strong increase in spring and then a certain collapse, because when grazing starts hitting, and that you get a net loss of the periphyton biomass. But this, of course, is always done from the perspective of a macrophyte. So what you need to have is data from studies that put kind of artificial plastic strips or whatever into the lakes to mimic new macrophytes coming or starting to grow in spring, which is then the situation which you have in most of the temperate lakes, that you have like annual macrophytes and they start to sort of grow and reach the surface where the light is in spring, and then you get typical periphyton shading patterns, and we normalized all these data to a percentage, and then you really get nice recurring patterns. So, and that's what we try to sort of mimic here. So in fact, it's the same what Annette already said, or Wolf already said. Also, it was already in the shading was already in the model, and now we take part of the periphyton of the phytoplankton shading away and put it to the periphyton where it actually belongs to.
Okay, thanks. Wolf, you had raised your hand. Please unmute yourself.
I managed to do it in one turn. Yeah, well, I was going to ask the same question as Annette, but that has already been asked. But I think one system that comes to mind where periphyton seems to be over important is the Everglades in the United States, which seems where most of the primary production seems to be created by the periphyton. Actually, Annette and I have been there canoeing, and so this is a huge system that seems to be completely driven by periphyton. Primary production by periphyton.
Okay. More questions to Alena. Okay, is not the case right now. Then if there's anybody who has questions to my presentation, I would be the next in turn. So, you see I'm kind of moving towards broader and broader scope in modeling, if you will. Annette and Wolf clearly top of the food chain here. So I'm ready to take questions from you now if you want. Please, Manchi.
Yes. So also a more general question. I think your study is very, very interesting. And I want to ask, did you already link this study to some more practical questions in the eutrophication management, or?
Well, I can tell the background of the second part of the talk where I was talking about these hypertrophic ponds. That was very much linked to a management issue because these hypertrophic ponds they have become something like a bird sanctuary where lots of waterfowl are just thriving. And there the question was how to best manage these ponds in the future to benefit the birds without, in a way, putting the water quality at risk. One thing that was foreseen for the future was that the wastewater that goes to this pond system is already treated, and in the future the plan was that denitrification step should be introduced maybe, and so that the reduction of nutrients going to these ponds would be a big thing, and the people who are trying to manage the system for the birds, they were actually starting to get worried because all these nutrients are beneficial to the waterfowl as long as you have relatively clear water, or even in some parts where you have turbid water, you benefit other birds. So in that sense, it was relevant. But it was actually not until I came to Umeå almost 6-7 years later that I started to link this to the model that we had developed for the benthic algae. So in that sense, there's no link from this model to any management, so to speak. So the management is more based on experience with that particular system, and I'm not involved in it anymore. So I was just advising some students who worked there. If that maybe not a satisfactory answer, but that's the answer I can give.
Thanks.
Okay, then I think Koka was, and then Sabine. Please, Koka.
Sebastian, you have shown in one of your slides the biomass of pelagic and benthic primary producers along gradients of background light attenuation, nutrient concentration sediments, as predicted by your very nice model, by the way. it called my attention the small area of the parameter space where alternative stable states can occur along these gradients. This suggests to me that this feature of shallow lakes dynamics that has been extensively explored in theoretical models have been oversold. I mean, do we have enough theoretical and empirical evidence to support the idea that alternative stable states are really a common feature of shallow lakes, or have we overestimated their importance? For instance, if we include more complexity to the model, such as the shape of the lakes you have mentioned in your talk, or the spatial heterogeneity of water and nutrients in flow that Annette has mentioned in her very nice talk, we find that this interesting type of dynamics disappears in the model, not to mention other complexities such as pulses of disturbances, etc. So this is just a question to you, but other people might answer too.
Yeah, maybe I can start because what, of course, I have to consider is that the figure I had in my talk is just one realization of possible parameter combinations. And an assumption we were making here is that the benthic and the pelagic algae would be slightly in their traits that would be different in a way that favors actually their competitive abilities, so that the pelagic algae would be somewhat better at taking up nutrients, whereas the benthic algae would be slightly better at taking up or using light when they are at the bottom. That was actually something I didn't want to have in the model originally. I wanted to have them identical because we get very similar results. That would have been much stronger in a way to show that coexistence is possible, and all these other things, albeit you make identical assumptions about the traits just by separating them in habitat and making this spatial asymmetry. But the reviewers and editors wanted us to do it that way. But we explored also other parameters, and you can actually increase the region of alternative states, but then often you have to make assumptions about the parameters in the model, like how much light is being absorbed by unit biomass and so on, that are maybe a bit at least against our intuition. So if you make these assumptions we made there, then this region tends to be fairly small, but if you make other assumptions, it can be much larger. So it's not one simple answer, so to speak. But yes, I agree in a sense. But on the other hand side, you also have to look at this figure from the perspective of, what kind of range we're using on the x-axis. It's like 1,000-fold range of sediment nutrients. You can actually take away at least one order of magnitude from the left side there. I think you will not find that in nature. So then, relatively speaking, the region already gets a bit bigger. So it's yeah, just so you know. So maybe that's my answer to that. But there may be other comments. I think. Well, Sabine had a question, but maybe someone else feels enticed to answer. If not, I think maybe that is good for the moment. Okay, so then let's take Sabine's question, and then we have to go on with other presentations. Yes, please, Sabine.
Sebastian, thanks for combining this with the fish study, fish pond study. I was surprised to see that your view data fit so well, because I was wondering in how far these floating algae ever covered that much of the surface that they could really prevent the phytoplankton underneath, because what you usually need with like a duckweed cover, so you need pretty much 100% coverage to at all limit the phytoplankton growth. So if you only have like several or like a certain percentage covered, which the photos seem to indicate, then I wonder how far really the phytoplankton was at all limited by light.
Yeah, so unfortunately, at the time when we were sampling these ponds, we didn't have these ideas. And we never took any light measurements and things like that. And also you're right in that, the ponds were at times almost, 100% is maybe never really possible, but on days that were totally wind still, you could have a pretty huge cover everywhere. Of course, that's something in reality these algae don't start out as floating mats. They are often initially benthic. So in the spring they develop and then they float up. So it's only a seasonal phenomenon. I have really no way of knowing whether this, in a way, it's an ad hoc hypothesis. At least at the time it was when we analyzed these data. That why do we get this negative response in chlorophyll? Because of course there's other things that also need to be known, but they didn't really differ. So this is that there were no fish in those ponds, which means maybe the grazes were developing, but we didn't really find any very significant differences, and grazes were not so abundant, and they would have been I mean, the absence of fish was everywhere, so that can't really grazing by Daphne and so on cannot necessarily explain why there was less chlorophyll in the ponds.
There is a nice study by Carolina Trochine in the Danish mesocosms where she showed that these floating filamentous algae can have allelopathic activity towards phytoplankton. Yeah. Maybe it's a nice one to consider here in your case.
Yeah, no, that so for that particular case study, I think you can easily come up with alternative explanations that we cannot exclude. I would simply say. So from my perspective, it was more like once we had this model for the benthic algae, where we pretty much know that things are probably working like that, even though we don't really know how much nutrients really leach out of the sediment, I have to say. But then it was like a eureka thing for me to realize, oh, I can just use the same model for our other system, and it seems to work, but it's only a hypothesis. So we have very little strong evidence that can really exclude alternative hypothesis, I would say. Okay, so maybe we have centered enough around my talk now. Let's go on to Annette's talk. Any questions to Annette's talk, please? Okay, maybe I start with a question. I was a bit wondering that Sabine might have asked it, because Sabine, you remember more than a decade ago, I reviewed a paper that was published in Oikos, where you were actually modeling a similar situation, not using PC Lake, but a much simpler model, in part also maybe a bit more phenomenological in some places. But what I remember was, and that confused me a little bit, one of the results you pointed out, Annette, was that you could get alternative states only in the situation where you had both seepage lakes and diffuse nutrient loading, basically where you were creating an homogeneous system. And I'm pretty sure that you got a different result, Sabine, and it makes sense to me that if you have your sequence of lake basins or whatever you want to call it, you must still be able to get an alternative state, at least in the most upstream system, under the conditions you would get it if the whole system was well mixed in a way. And what I remember from your paper was that under those conditions, you could have actually a situation where this alternative state could be propagated downstream to some extent. And I'm just curious, why are you not getting this? Is this a matter of parameterization, or are you sure that it's impossible?
Actually, I get this. So this is, I think, the lake type where you have like water as a point source and nutrients at diffuse source, if I'm right. Yeah, I think it was.
No, sorry, I think, well, maybe I'm wrong. I don't remember that paper, but I'm thinking point source both. Everything comes at the upstream end. And let's assume it's a situation where you get an alternative state in this upstream end. Why doesn't it propagate downstream to some extent?
Well, I have a lake type, but then I have to go back to my figures, which one it was. But where you have this propagation of alternative stable states, which you can see from the figure, where you see like small humps in the figure, so. But no, then I have to get the figure back.
I don't know. I'm bringing up your.
It's a diffuse loaded drainage lake. You see, where you have like a little bit difference between the solid line and the this dashed line.
Yeah, this one.
And
I
there
guess
you
where.
See this.
Yeah, but I was actually wondering about the point loaded drainage lake. Shouldn't it be possible there also?
Yeah, that's a good question. Maybe, so here I took the average of all the lakes, and I think Sabine didn't take the average of the whole basin, but looked at the different parts of it. So then we have to go into the details of each of the lake part, and then I think it's the case, because then it just is the first one is just simply a PC Lake model, and it's not affected by anything else. So then I think there is locally there is an alternative stable state.
Yeah. Okay.
Yeah.
And then I had one second question about this, and this is since you introduced this with this large Chinese lake where you have different basins that look differently. The assumption you're making in this let's say a chain of lakes or lake basins is a very strong one, which is that you have a unidirectional effect. Obviously, in a real lake, you would have some effect also in the opposite direction. And I think it's very useful to look at an extreme case first. But have you explored, how much of a let's say diffusive movement or movement in the opposite direction is necessary to break down certain patterns or so?
No, I didn't look into it, but I would expect that it would make it more homogeneous in that sense.
Yeah.
But yeah, so you have, of course, diffusion, or you have really the water flow that goes the other direction. Diffusion, I would expect to have not so much effect because it's a slow process. So then we're talking about maybe the water flow itself, the advection. And that I would think that it would make it more homogeneous. Yeah.
Okay, Wolf has raised his hand, and I ask him to unmute.
Yes, I managed to do that, Seb.
Yes.
Well, it's more, may I re-ask Koka's question to you specifically, Annette? Do we overestimate the I mean, Seb has this nice mechanism, it's very sexy that the thing goes down when something goes up and everybody's surprised and you can publish it nicely. You're absorbed by the phenomena. And are we all looking like rabbits in the headlights of the alternative stable states, whereas the real world is around us, is differently? What's your opinion about that?
Well, the longer I look at it, the less I see them. So in that sense, I would say they're less common than you would expect. At the same time, I think it also depends on which scale you look at. I just said, like, I took the average of the entire lake where there's spatial heterogeneity, but I think if you really look to this first part of the lake, I think you will find it. Because then it's just a simple PC Lake, and PC Lake has it. So I'm not sure how that works in nature. If you would zoom in nature, if you would then go to a really small scale, whether you also would find it, that I don't know. Yeah, but
yeah, I can maybe add from my own experience that the lakes we were looking up here in the north, they would be definitely in a range where I don't expect alternative states, at least not through those mechanisms. There could be other things happening, but it could be maybe that state-structured fish populations could cause alternative states or so, but probably not these mechanisms.
So the lakes here in Brazil we study, Sebastian, they are outside the range of this nice and sexy type of dynamics, as Wolf said.
Okay. More questions? I think I have to push, actually. We are already 8 minutes to 4 and on my local time here, which is you guys in Brazil are getting hungry, I think. So Wolf is the last presentation we have left. So are there any questions to Wolf or his presentation? If this is not the case, then maybe I again kick off something here. So what I've been thinking, and that is something I think a bit generally, is that because Wolf, you're in a way suggesting that we need to expand the scope of our models even more, or at least our vision, what we take into account, which could be some things like evolution, new species, and so on. Of course, this will all increase complexity even further in many senses. There's always this, let's say, this tension between understanding and prediction, for example. And I think we've been discussing it a few years ago also. I'm more the kind of researcher who just loves to understand things, and then I'm happy with it, which is maybe totally irresponsible because we have to deliver to the society. But this is I'm just a kid playing, and it's so much hard work trying to understand things that once I get something out of it, I'm happy enough. Whereas people who deal with lake management and so on, of course, have other claims on them or expectations. But the thing I'm actually wondering is that since several of you have used in their research a PC Lake, which is a very complex model in many respects, as we have already seen, and still has limitations in the sense that some things are not really mechanistically included there. So I'm actually wondering how much of the phenomena you are seeing and you have been presenting, which a lot of it was about these potentially alternative stable states. How much of that depends on what are your assumptions that you're dealing with in the PC Lake model? For example, is it a model version where you have piscivorous fish in, or is it one without, and so on? Because I know from my much more simple-minded modeling that I get totally different results with my benthic pelagic algae model if I include grazers. And then if I move up and include fish, I get yet again different results. The question is then whether using a very complex model as the background in which you explore these more specific questions, whether you are getting a full understanding of the phenomena, or whether you're actually just looking at a slice that is very much contingent on what you assume in the remaining model, which is the background. So I'm just kind of throwing this up as a more general question.
Yes, I say something about it. I already unmuted, so that's good. Because you were talking, I had to do it myself. What I'm thinking now about is that I fully agree with you that the level of complexity in part determines the phenomena that we observe, right? So if you throw in more complexity, you get different phenomena. And if I see your models anytime, as much as I like you as a person, I don't like the level of complexity that you work on. All the field times, you should add a little bit more this and this. So my completely subjective comfort zone of complexity is around PC Lake, and yours seems to be around this kind of more. I'm rereading Don Diense's book on nutrient cycling at this moment. It's completely your level of complexity, I think, that you feel comfortable with Seb. So the little bit more mathematical, focused on a certain food feedback loop, and there's loads of stuff to study there. And actually, what I propose here is, with the co-authors, of course, Dianneke was heavily involved, and Sven and Mansi and others, is that in a changing world, all of us think about adaptation, and organisms have Darwinian mechanisms of adaptation, and they have phenotypic plasticity, and we have all these invasive species, and humans, we try to adapt. So there is all kinds of responses to global change and local change. But we make models like they are clockwork. We live in the Newtonian world with our models and with fixed parameters and so on, and that doesn't match for me. But what I propose is far outside my own comfort zone of complexity. So I propose it, we propose it, but I don't do it because I don't feel comfortable there at all. And I think we completely lose sight at what we're doing and then wouldn't understand. So I'm on what you feel maybe for PC Lake is what I feel for these models, very uncomfortable and unpredictable. But still, that then means that we only study what we feel comfortable with. Whereas I'm sure that we see in a world that is changing. I mean, the whole thing is about global change. Why are we worried about it? Because it forces adaptive responses in societies and in communities and in individuals that are not built in because we never experienced it before in in our history, right? And neither natural history or the human societal history. So should we ignore it because we don't like it, or what should we do?
Yeah, I actually, I hope you understood that. I'm not criticizing this approach.
No, not at all.
No, it's more that, what I'm sometimes think about is, we know, for example, climatologists who want to make predictions about how global climate develops. They use extremely complicated models, and I don't think any of them can explain why a particular model outcome arises in their model in the way we at least I often would like to understand to pinpoint certain mechanisms that have certain consequences. And I don't think that among us scientists there are many people who would, how to say, these models. You know, they're extremely useful, these predictions for the future. Of course, we see there's a lot of uncertainty and so on, but and they do these, I guess, ensemble models, what they call them, or so. So there are 15 different high-tech computers in different countries who run their own things, and then they put it together and somehow weight it together. And this may be a way that is the only one that works at this stage in order to make predictions for the future. And these predictions, I think, are very useful and needed. And without them, we wouldn't get the public pressure and the politicians to realize that things have to change. So that's pretty clear. But I guess on the lake ecology side, we are not really there, you know, probably. And I don't know if, you know, in meteorology, for example, there's no question that there are lots of people working this way, simply. Whereas,
but Seb, that's also a timescale issue then, because the crisis is here. So if it takes us 30 years more in limnology to solve the problems that are there tomorrow, then there is a timescale issue with the scale at which our science develops and at which the crisis develops.
Yeah, no, I'm more thinking about, are we there where we even can make reasonable predictions with the modeling tools we have now? That's more what I'm wondering. If we had like 15 different lake models and so on, and I guess they are there in a way, at least in certainly in the marine realm, there is all these, you read the acronyms, and that's usually models where you need a phone book thick of explanations how they work. PC Lake is a dream compared to that, I would think. Yeah, there's all these details built in, and you can't really understand them anymore. But I'm not sure whether we are already there for lakes anyway. You know, that we have these multiple tools that we can weigh against each other.
Coca raised his hands already a long time.
Oh, sorry. Yeah. So please.
I think these very interesting discussions remind me this very well-known trade-offs between realism, precision, and generality in model building. We know it's just impossible to maximize the three of them. And what we use models for to explain and to predict. And a model by definitions are wrong. And you know, we need to abstract from reality things that we think might be important to explain some phenomena of interest and leave a lot of other things out. And this is very important. This is very critical in our work with modeling. But I think, to me, one very important thing is to keep the model as general as possible. Because to me, science is about generality. I mean, we need to make inferences about many cases and not just a single case. Because if you have a model of a single case, we don't have a model have just the description. And then I believe to keep the model as general as possible, it should be a major goal in our modeling effort. And then, but on the other hand, I've been very much worried about my lack of knowledge in economics, for instance, in social science. Because to me, the causes for water management problems, for environmental destructions, are always has an economical basis. And to put inside our model some important economical variables, socio-economical variables that we need to apply our models for management, it's tricky because we are not, we don't have the background. We need to cooperate with other people from other fields, starting from hydrology. I mean, water is a resource for any kind of economic activities, and we need to include this hydrology in our models and some socioeconomical variables in our models if we are to use them for management purposes with efficiency. I'm not personally, I'm not so much interested on this eco-evo dynamics, even though they are very important, for instance, in the climate change issues. But I'm more interested in this socio-ecological systems. But my problem is, I mean, I feel very ignorant on the social sciences to be able to do it by myself. So that's my impression.
Thank you. Annette, I think you raised your hand also.
Yeah, so I think also in response to you, but also I agree with Koka that so a model should suit its purpose. So I think the simple models, you can I think it's easier to understand at least than PC Lake. I wouldn't say that it's necessarily easy, but easier than PC Lake because there's a lot of things going on in PC Lake. But maybe with PC Lake you can do other things than the simple models. At the same time, they also sometimes give the same pattern. For example, Luc van Gerven once studied the effect of hydrology also and nutrients in a ditch system. And then he found similar patterns in simple models and complex models. So I think there we can see how they diverge. And then I wanted to add, so you asked about whether we should do ensemble modeling with the lake people as well. And I can add to that so there is a project called EasyMap. It's a project worldwide going on, and they aim to use different kinds of models to predict how the world will respond to all kinds of things. So there are different sectors, but one of the sectors is a lake sector. And there they already did an analysis on the water temperatures in lakes around the world. And they really want to go also to the water quality indicators, such as algae, phosphorus, nitrogen, this kind of things. The issue is here that we indeed, like Koka said, like we need to connect to, for example, hydrology or to also nutrient flow models to really make this effort being done. But these developments are there. So it's not possible, I think, this year, but I'm sure it will happen somewhere in the future. So yeah, I think if you're interested, you can join this this group.
Okay, yeah. Koka, I had a spontaneous thought. You know, when you were starting to talk about these, let's say, socioeconomic factors also that that are involved in lake management and so on. Of course, I fully agree with you that the most general model is preferable in a way. But of course, I would still see that there would probably be regional differences in certain, you know, especially when it comes to socio-economical factors. For example, in more arid areas, of course, that demand on water as a resource is extremely high. Whereas if you go to the subarctic in northern Canada, nobody cares about the water that's there because nobody's there that uses it and so on. So maybe in those areas you have to worry much more about forestry or so, you know, how they handle these kind of things, which you may not have to worry about in other areas, which are purely agriculture and so on. So of course, the socioeconomic side adds also diversity to the modeling in the same way as if you go from an oligotrophic to eutrophic lake, or from a tropical to a temperate lake, where you have different organisms and these kind of things. So you once you want to apply this to a broad range of lakes and including their catchments and so on, I guess you still have to make decisions on which things to exclude at least and which things to include. While you could still have a very general template in a way. Let's put it that way. Wolf, please.
Well, it's just a closing remark. If you would give me a few seconds at the end of the session, I would like that.
Yeah, okay. Yeah, so as I said, especially the Brazilians are probably really hungry. So I think we don't want to extend their suffering, or not only the Brazilians, but everybody who is in that time zone, let's put it that way. So maybe I give Wolf the voice to have some concluding remarks on this session. And I already thank you all for participating. Please, Wolf.
Well, this is, of course, an adventure for all of us with the conference. I'm really impressed by the way the conference is organized by the Brazilian side. And I think in person of Coca, I want to thank you for that. And also this session, then the work that Seb put in it and to prepare it and to structure the time. I mean, I love to be with you in the same room and have a beer afterwards and everything. And the whole chemistry is different. But in terms of scientific effectiveness, this was as good as it gets, I guess, in terms of a session. So I'm really impressed by what we can do on the scientific side. We missed the social side to a large part, but scientifically, I think both the conference so far and this session was a great success. So I want to applause for all of people.
Yeah, I just can't agree, and I would love to give you all a big hug. Those who know me, you know, you wouldn't escape, but maybe there will be opportunities in the future. And thanks for joining and contributing. I was very stressed this time, I have to say. You know, it's the worst time of the year for me to be in a conference. And I know Coca had some sleepless nights because of me not responding and so on. But I guess we kind of pulled it off. And thanks to your great contributions. Okay, I look forward to interacting with you in other sessions in the rest of the week.
Sebastian, just a final word. I wish on behalf of the organizing committee, you a great day, and thank you very much for being here with us. We are very glad that everything worked fine, smooth. We were very worried about, because I mean, we're learning by doing. We never done such a thing before. But I think the session was great. Thank you very much, Sebastian, for leading this for us. I was indeed worried because, I mean, you've been so busy and teaching a lot, and you didn't respond my emails. And I was wondering, does he know that tomorrow is his section? but you did great. Thank you very much. Thanks for all of you.
Yeah, and then maybe Carlos and Coca, we can make up for another meeting and talk about the detailed questions I had about your presentation. But that's maybe next week or so, when everybody has recovered. Yeah.
Thank you very much.
Good. So thanks a lot. I will leave this room now, but I hope to see you in other rooms in the future. Okay.